All right, well, first, good afternoon, everybody. I'm Cory Inman. I'm an assistant professor in the Department of Psychology at the University of Utah. Thank you, Rodrigo, and to the organizers for having me. Today, I'll be talking to you about modulation of emotion and memory via direct brain stimulation in humans. This is about the last 10 years of work in my postdoc and when I was starting my lab. So I'm going to try to fit it all into this next 15 minutes or so, so bear with me a little bit. So first, I want to say that I'm just here as a representative of a great group of people that have really helped me learn how to do this work, from the folks at Emory and WashU to my current lab, who's really the folks that are doing all of this work, and I hope that you'll get a chance to meet them one day. So this is just an outline. If you want to take a picture of it, this is a good place to get your phone out, take a picture. This is what we're going to be taking home today in terms of the messages. Like I said, this is about 10 years of work that we've been working on doing the same, very similar task in lots of epilepsy patients. I'll come back to that later. But we're essentially going to be starting with talking about the sort of autonomic responses and subjective responses to amygdala stimulation in a cohort of patients that we tested early on to try to figure out whether this was a safe thing to do theta burst stimulation, and what the sort of safety limits were, and to also understand the extent to which this modulates subjective experience and physiological experience. Then we'll talk a lot about how amygdala stimulation affects memory, specifically recognition memory, and our experience with that. And then we'll talk finally about some of the stuff that we're doing for our future directions. All right, so first, autonomic and subjective experience, subjective effects of amygdala stimulation in humans. One, we know that the human amygdala mediates our emotional response. We've talked about it today. We've covered this in lots of cognitive neuroscience work. And we know that these emotional responses involve changes in three primary domains: the sort of neural response, the behavioral or the subjective response, and the physiological response. And so we wanted to ask the question in this first work of are changes in each affective domain mediated independently by the amygdala? In other words, could you see one of these effects without having to see all of these effects? And so we did a very simple experiment. Like I said, we're still exploring the sort of safety limitations of doing amygdala stimulation, because amygdala stimulation can provoke seizures. So we wanted to stay within those bounds. And what we did was we recorded electrodermal activity, heart rate, and respiration as we did a stepwise paradigm stimulating progressively more higher amplitudes of stimulation. We mixed in randomly, we just mixed in some sham stimulation where we pretended to turn it on. We would say that the stimulation was on, but we weren't actually turning it on to get rid of any, or to be able to control for any sort of expectancy biases. And so the initial work in this just shows kind of what you would expect, that the higher the amplitude, the larger the physiological response we see. The key here is that all of these physiological responses were made without any sort of subjective experience. So this was 7 of our 8 patients showed a clear skin conductance response. This is just an example, one example patient. And then over here we show that you see this same sort of trend of a dose-dependent response for every one of our patients when we stimulate the amygdala. And you get this bigger skin conductance response. And just to point out, this is 10 microsiemens. This is a very large skin conductance response. We also, interestingly, see a decrease in heart rate. We see a bradycardia. This is the same kind of bradycardia that you would see in kind of a defensive freezing state that's in more of a preparation for activation of the fight or flight response. So it's a sympathetic response that you see early on with heart rate. Now, that being said, we did have one patient that showed a very stereotypical response that you would expect, but it was only in one of our patients. And so if you go back and read that paper, the story I would like you to take away from that is that most people actually, in fact, do not get any sort of fear response when they have amygdala stimulation, even at these very high doses. But we do get subjective effects, and this is one of the examples. If we could make sure that the audio is on, that would be great. You can also get what it's saying at the bottom, but it's much better if you can hear it. It was working in the other room. Yeah. So I'm gonna. Can I pause things? No.
It was terrifying. It was like I was about to get attacked like the moment, like someone unleashes the child on you, and it just like gets so close, and you feel like you're gonna shit your pants. It's terrifying. It's not pain, but just fear.
Found that quote particularly interesting.
Is it interesting?
Fun or fun?
Fun? Okay.
He found it to be like kind of like riding a roller coaster. Like he knew he was safe, but he was.
It was ziplining a few weeks ago,
Okay.
and this was worse.
So he was ziplining over the 1,700-foot gorge in Colorado. So yeah, he had a very explicit subjective response. We actually replicated that a week later as well, and he had the same response. We also made sure to check in with his wife to make sure she was okay that we were working with him this way. We kept checking with him, make sure he was okay with this. But yeah, he had a very vivid subjective response. It seemed to be a little bit different in nature, also in terms of the physiology, in that he had same sort of increases in skin conductance, but he also had increases in heart rate immediately, which is much more reminiscent of kind of an immediate sort of fight or flight response. And so we can see that here. So at the higher doses, he was showing these kind of immediate kind of increases in heart rate rather than an initial decrease in heart rate. So we thought of the stuff that one of the reasons we think this was the case is that we, in this case, we were stimulating closer to the central nucleus. We were more medial, which would have more projections to the hypothalamus and then out to the cortex. Whereas in most of those other patients, we were actually primarily stimulating the basolateral amygdala and with more projections to the medial temporal lobe. Speaking of the medial temporal lobe, now I'm going to tell you a little bit about the work that we've done showing that direct electrical stimulation of the amygdala can enhance declarative memory in humans. One of the things that my lab is very, very interested in is how do we remember the first-person experiences that we have every day, the sort of autobiographical experiences. How are you going to go and take away different things from this meeting, right? Why do we remember some of the certain things that we remember? And we know that emotional events often stand out in our recollections, and there are whole animal and human studies and whole literatures that implicate the amygdala, and in particular its connections to the hippocampus. There's whole movies about this that discuss why emotions help us make memories. And so we're building this work on a very rich literature, which is a bit different than a lot of the other sort of approaches to doing brain stimulation for memory enhancement. So the basolateral amygdala specifically is known for its modulation of memory via interactions with other brain regions, specifically the hippocampus and other adjacent medial temporal lobe structures. And if you do optogenetic stimulation of the amygdala in animal studies, you can stimulate the amygdala. That provokes changes in not only oscillatory activity, but it also upregulates gene expression, or early gene expression proteins like ARC protein, that is known to be involved in the sort of consolidation of memories over time. So it strengthens those memories with longer periods of time. It sort of initiates this cascade of long-term potentiation and consolidation. And in humans, we have the opportunity to work with epilepsy patients like you've heard about today. I won't be telling you guys anything about single units today, but I will point you to a recent paper that we have on single unit responses to amygdala stimulation. In this case, it's theta burst stimulation that is now out in eLife as a preprint, and we'll be submitting the final manuscript of record soon. So if you're looking for responses of single units to amygdala stimulation, you can find some more work there. So in this case, we're going to use direct electrical stimulation of the amygdala like we talked about earlier. In this case, it's theta burst stimulation to try to mimic the sort of endogenous oscillatory activity that the amygdala and the hippocampus seem to run in. And we're going to then look at some of the electrophysiological changes that we see to that. And I'll tell you about a little newer work following up on this. This is our patient's experience. That was supposed to be our patient's experience. Essentially, they're sitting in a bed and they see objects on a computer screen. We have 14 patients in our initial study that had electrodes in their basolateral amygdala. We localized these to the basolateral amygdala by stretching a atlas on top of their own physiology and on a T1. And then we made sure that all of these were kind of hitting these areas prospectively before we went and did our tests. In this experiment, we're just showing one picture at a time. These are just neutral objects that are presented one at a time for 3 seconds. Then right after, they get 1 second of theta burst stimulation, 8 hertz, 8 bursts of 50 hertz in 1 second at about 0.5 milliamps. And we're doing this stimulation afterwards so that we could control for any sort of attentional effects that we might have. This is also what we had done in prior animal studies, is stimulate right after the animal had explored the object. And then we're just asking them to orient to these pictures by asking them whether this is something that they typically encounter more indoors or outdoors. At the immediate test, we mix in some new items along with half of the items that were stimulated and half the items that were not stimulated, and then test their recognition memory for whether this is a repeated or new item. And then we do the same thing at a 1-day delay to look at changes over time in terms of consolidation. We also wanted to make sure that the patients weren't feeling any of this. We purposely picked 0.5 milliamps because we knew that would stay under any sort of subjective awareness threshold. And this is a silly figure, but we confirmed that we don't see any sort of subjective effect. Nor do we see any sort of physiological effect. We don't see any changes in heart rate or skin conductance at this level of stimulation. So what did we see at our immediate test? We saw this kind of mixed bag effect where about half of the patients showed a memory enhancement effect. That's any line that is going up here from the blue to the red. We saw a couple patients that actually seemed to show somewhat of an impairment at the immediate test, and some people that just didn't show much of an effect at all. But then after the night of sleep and after about a day, we can show that 11 out of the 14 patients showed a memory enhancement. We can depict this in a slightly different way here, just subtracting the position of the non-stimulated dot to the position of the stimulated dot. And you'll see that we see a very consistent effect in this first data set. Operative word is first data set. And that's because we kept doing more experiments like this, manipulating some of the parameters like the timing of the stimulation, things like that. Those don't seem to have much of an effect. But the good news is that we overall continue to see a similar sort of memory enhancement effect. The difference is that we start to see some patients that show an impairment. And so we have spent a lot of time over the last 4 or 5 years trying to figure out what is leading to these patients that are having an impairment, and sort of spreading this distribution out. This is not too surprising. This is a fairly normal distribution that is still a significant effect above 0 here. So we spent a lot of time looking at individual differences in where exactly we implanted the electrodes, all of those sorts of things. So this is all of our patients kind of in the order that they were tested. You can see that as soon as we start manipulating some of the parameters that we start to get some impairments. Another thing that is critical here, and when we have a paper in Cognitive Affective and Behavioral Neurosciences that from last year that shows that really the thing that is most predictive of whether someone gets an enhancement or an impairment, in other words, that they're susceptible to some sort of memory modulation, is whether their baseline pre-surgical neuropsychological measures of memory were high or low. So if they had good or normal memory to begin with, then they were not susceptible to any sort of memory modulation, either an enhancement or an impairment. This speaks against any sort of kind of interpretation that would suggest that this is just a ceiling effect, right? Because even the people that had good memory to begin with didn't get impaired either. And so you can see that the darker dots are all people that are either having a pretty big enhancement or a pretty big impairment, and that a lot of the brighter dots lie around 0. So that's an interesting data point. It is both useful and challenging if we were to try to use this as a technique to help try to enhance people's memory clinically one day. And it's one of the challenges that we're trying to face right now, to figure out exactly what the differences are between the people that have a memory and impairment and the people that have a memory and enhancement. All right. So in terms of electrophysiology, what we see is that we see an increase. We had 5 patients that had coverage of the basolateral amygdala, the pararhinal cortex involved in object memory, and the hippocampus. We see between 30 and 55 hertz an increase in power, specifically in the pararhinal cortex, which has been implicated as being very important for object memory. We see an increase only for the stimulated items, and this is remembered stimulated versus remembered not stimulated. So the only difference here is that these items were either stimulated or not stimulated. The other key here is that we're actually looking, we couldn't look during the actual memory encoding, so we had to look during memory retrieval. So this is when they're actually seeing the picture the next day, one day later, that you get this echo of the effects of stimulation the next day tagged to specific items. And then we also see some interesting patterns in terms of the relationship between the amygdala's theta activity and pararhinal activity in the gamma range, where the gamma activity happens at a specific phase of the amygdala's theta. So you can see that here in terms of the modulation index. Interestingly enough, this actually mimics, to some extent, this kind of 8 by 50 hertz pattern that we introduced initially. I'm going to zoom through a couple new findings for sake of time. But in this next experiment, we were interested in whether amygdala stimulation would also enhance memory for scenes or not enhance memory for scenes. And our hypothesis was that it would primarily enhance memory for objects given the anterior hippocampus's role in object memory and the posterior hippocampus's role in scene memory, and that there are more dense projections from the amygdala to the anterior hippocampus versus the posterior hippocampus. This is a recent preprint that's been put out. We scrambled images of the scenes to put behind the objects, and so that they all had similar amounts of information in them. And for objects, we replicate what we've seen before, where we see memory enhancement consistently after a one-day test. And then for scenes, we show that you actually see a bit of a mixed bag. If we display this in this kind of difference measure, you can see that, in fact, some of the patients actually show a memory impairment for scenes, while at the same time showing a memory enhancement for objects, which is an interesting way to sort of dissect this circuit. Going back to the sort of single pulse electrical stimulation that Peter was mentioning, we also can map that and use that as a way to sort of interrogate this circuit to see is the extent to which there is effective connectivity between where we're stimulating the amygdala and the anterior versus posterior hippocampus. Is that consistent? And yes, we do see that there is a larger sort of single pulse effective connectivity response in the anterior hippocampus. So I'll leave this up briefly. These are all the things that we think are unique about amygdala stimulation for memory enhancement. And essentially what we think this is evidence of is causal, that direct amygdala stimulation causes prioritization of temporally specific declarative memories for later recognition memory without eliciting any sort of emotional response. And then we hope that with further tuning that this amygdala-mediated memory enhancement can maybe a novel therapeutic approach for memory disorders. So if you want to learn more about this, come chat with me later. I'll tell you about it. And thank you very much.
Thank you, Cory. Unfortunately, we have to move on for time, but thank you.